With this, let us now start the most important part that of Plasmodium, that is laboratory diagnosis. After so many years of diagnosis also, after so many years of discovery also, still then they say that smear microscopy is the gold standard method, peripheral smear microscopy. Peripheral smear microscopy is still the gold standard method. There are two smears are available, thick smear and thin smear. Which is better? Which is more sensitive? Where amount of blood is more? Thick smear, the amount of blood is more, hence it is more sensitive. It is also used for quantification. For quantification, thick smear is the best method. It is also used to demonstrate malaria pigment. It is also used to demonstrate malaria pigments, whereas the only advantage of thin smear is it is used for speciation. For speciation, intact RBCs are required, and thick smear, the RBCs are lysed. When you make the thick smear examination, while you prepare the thick smear examination, the RBCs will be destroyed, hence the intact RBCs will not be there. For speciation, you need intact RBC so that you can know which species is there inside. So, now I will tell you how the speciation is done, how the thin smear is used for speciation. See, speciation is done based on 5 important properties: the type of ring form, the type of gametocyte, the type of sygont, the malaria pigment, and the RBC structure. Depending upon all these 5 properties, the speciation is done. Okay. So, let us discuss one by one, let us discuss in a reverse order. First, I will tell you about RBC. RBC, they say that for Plasmodium vivax, RBC is enlarged. For oval, RBC is enlarged, oval and irregular margin. Enlarged, oval in shape and having a irregular margin. Whereas the remaining 2, that is what? Plasmodium falciparum and malaria, RBC is normal. Coming to malaria pigment. Malaria pigment, they say that for all species, the malaria pigment is dark brown in color, whereas except Plasmodium vivax, where it is yellowish brown in color. By detecting which color of malaria pigment you can speciate. Sygont. Sygont means what? Sygont is a big sac-like structure having merozoites inside. How many merozoites they have? This is a property by which you can speciate how many merozoites are there inside. For vivax and falciparum, 12 to 24 merozoites per sygont, whereas malariae and oval, there are 8 merozoites per sygont. 8 merozoites per sygont. And one more thing I want to emphasize here, falciparum in the peripheral blood smear examination, only ring form and gametocytes are seen. Okay. The other forms are seen only in the deep vessels, not in the peripheral blood vessel. So, this is again a point to be noted, because falciparum usually multiply the RBC cycle, the most of the part of the RBC cycle occurs in the deep organ vessels, brain vessels, liver vessels and all that. Peripheral blood vessel, only the first form and the last form will be seen. First is ring form and the last form is gametocyte. The remaining stage occurs in the deep vessels. That means if I ask you if you have 12 scion in a peripheral blood smear examination, it could be what? Which species it is? Falciparum or vivax? 12 to 24 is either falciparum or vivax, but in peripheral blood smear only vivax is seen, falciparum is not seen. Next is gametocyte. Gametocyte, they say that falciparum, the gametocyte is banana shaped. Banana shaped gametocyte, banana shaped or crescentic gametocyte. Whereas other non-PF, it is round gametocyte. Gametocyte is round. Then the most important part, that is ring form. Very frequently asked MCQ. Ring form, based on which, how can you differentiate? Ring form of vivax. Vivax, what is the shape of RBC? RBC in vivax is enlarged. Enlarged RBC. Having a enlarged ring form. They say that the ring form occupies 1/3 of space of RBC. This is the ring form or early trophozoite of vivax. Whereas the late trophozoite of vivax, of course RBC is enlarged and the ring form, the cytoplasm will become irregular, called as amoeboid ring. The cytoplasm becomes irregular, called as amoeboid ring form. So this is about vivax identifying feature. Falciparum, the RBC is normal shape RBC, having 3 variety of ring form. One is it has multiple ring form. Other one is ring form attached to RBC membrane, called as ecoliform. Ecoliform is ring form attached to RBC membrane. Then one more variety is sometime double dot ring form will be there. The nucleus is broken into 2 pieces, which is called as double dot or headphone shaped ring form, like this. Double dot or headphone shaped ring form will be there. Okay. Coming to Plasmodium malariae. Plasmodium malariae RBC is normal small, I mean not enlarged, normal round RBC, and the ring form is not actually ring, it is actually band shaped. Band forms, they are called as band forms, or band shaped forms are seen in the, instead of ring it will become a band shaped. Oval, what is the shape of RBC? Oval, the RBC is irregular, oval and enlarged, it is oval in shape and irregular margin, fimbriated, having a ring form inside. So based on the RBC you can tell that it is oval. The ring form does not have any specific feature, but based on RBC shape you can tell it is oval. So this is all about peripheral blood microscopy. So other than that, other diagnostic tool for malaria, you can go for a fluorescent microscopy called as Kawamoto technique. A fluorescent plus centrifuge, which is called as QBC. QBC is quantitative Buffy code examination. Quantitative Buffy code examination. Here they say that the blood is collected in a capillary tube. blood is collected in a capillary tube, and this tube is coated internally with acridine orange, coated internally with acridine orange. This is a stain which is used here. So, once the blood is collected inside, the blood is subjected to centrifuge. So, when you centrifuge the blood in the capillary tube, the blood cells will be separated depending upon their molecular weight, depending upon the density the blood cells will be separated. First, the bottom zone will be the heaviest cell, that is RBC. Then you will have the lighter cells, that is WBC zone. Later on the platelet zone will be there. So, like that the blood cells will be separated. Then you have to examine the Buffy coat area. Buffy coat area is examined. Buffy coat area means what? Buffy coat area is the junction of WBC and RBC. Is the WBC junction with RBC, the junction of WBC and RBC, this is the Buffy coat area. This is the area where the parasitized RBCs will be there, and this is the area which is examined under fluorescent microscope. So, this is also fluorescent stain, but along with that centrifugation is done in a capillary tube, that is called as QBC, quantitative Buffy coat examination, and they say that it is almost equal sensitivity like thick smear examination. Of course, thick smear is best, followed by QBC, quantitative Buffy coat examination. Then antigen detection, third is antigen detection by doing immunochromatographic test. Here they say that you can detect various antigens like parasitic LDH, histidine rich protein 2 and aldolase. Among the three antigen, the LDH and aldolase antigens are pan-malarial antigen. They are present in all the species, whereas histidine rich protein 2 is PF specific. It is PF specific antigen. It is present only in Plasmodium falciparum. So, if histidine rich protein 2 will be positive, that means it can confirm that it is Plasmodium falciparum. Okay. Then culture, it is not done routinely, it is done only in the research center. RPMI 1640 medium is used, and by following a method called as Trager and Jenser method. Trager and Jenser method is used using RPMI 1640 medium. It is not routinely done, it is done only in reference centers. Fine. So, this is all about the, and of course PCR, the last diagnostic tool is PCR using the various primers, and PCR is most sensitive. It can speciate all the 4 species, as well as it can detect the drug resistance gene. It can detect the drug resistant gene. Hence, PCR is quite, but the thing is because it is sophisticated, it is very difficult to perform. So, it is again limited only to the higher centers. Okay, then a bit about Plasmodium knowlesi. Plasmodium knowlesi is the latest species discovered. It is a parasite of monkey. It usually infects monkeys. Not reported in India yet. Over the world, they say that around 2010 onwards, cases have been reported mainly from Thailand, also from other countries like Singapore and all, mainly from Thailand and also from other Asian countries like Singapore, etc. Here the vector is, they say that here how it is different than diagnostic feature. They say that Plasmodium knowlesi, the early trophozoites, resembles that of Plasmodium falciparum, whereas the late trophozoites resembles to that of Plasmodium malariae, band form like it will be there, which will resemble to Plasmodium malariae, whereas early trophozoites they resemble to Plasmodium falciparum, multiple ring form, other forms will be seen. Okay. And as I told you here, the vector is a particular species of Anopheles called as Anopheles leucospirus. And they say that though Plasmodium knowlesi is not found in India, but Anopheles leucospirus is found in the western coastal region belt of India. So, there is a high probability that we may get Plasmodium knowlesi in near future. Okay, and of course, fever I have told you, fever pattern is quotidian pattern every 24 hours.